Hello guys, today we're going to talk about the nerve system and the nerve tissue actually. So first up, let's discuss the basic organization and the difference between the CNS and the PNS. Well, CNS is the central nervous system and is composed of three different components: the cerebrum, which is the brain, the cerebellum, and the spinal cord. Now, when we go to the PNS, we'll see three different components, which are the cranial nerves, the spinal nerves, and the ganglia. Ganglia are typically just nothing more than aggregations of nerve cell bodies and cell processes of the cell processes, of course, of the nerves. So let's discuss about the nerve tissue. So in the nerve tissue, we have nerve cells. There are two different groups of nerve cells: the neurons and the glial cells. Well, the neurons are the cells that actually transmit and generate the action potential, of course, and the electrical signals, along with all the consequences of these actions. And we have the glial cells, which are the supportive cells. Now, the glial cells have many roles depending on their location, if it's at CNS or PNS, depending, along with many others. Let's see some of the basic functions of glial cells. Well, first off, there's the physical support of the neurons, the protection, mechanical protection. Then we have the metabolic exchange between the vascular system and the neurons of the nervous system. This will actually see many cells that focus exactly on this, that form the blood-brain barrier, the blood-CSF barrier, and of course these structures are necessary to maintain a healthy CSF and many other points that we'll discuss in a bit. The point is that because of the supportive cells, we're able to better regulate metabolically, along with also when it comes to terms of nutrients and oxygen, of course, between the nerve system and the rest of the vascular system. Of course, these cells also are responsible for the insulation of the null cell bodies. This is a bit special term. Why? Because insulation doesn't necessarily refer only to thermal, but it also goes to mechanical, thermal, and electrical isolation of the nerve cells and protection of the nerve cells. Of course, the glial cells also play a role in the repair of the neural injury, though it is true that the nerve system is way more fragile to neural injuries. Like the regeneration potential of nerves is not the best in the human body. Of course, there is some limited potential, but this is the most limited structure for when it comes in terms of regeneration, in terms of repair. Similar to the, of course, heart. All the other tissues in the human body have higher potential for regeneration. The nerve and the heart typically don't have these potential as the rest of the tissues. And last, we have the clearance of the neurotransmitters from synaptic clefts. We see the two terms, neurotransmitters and synaptic clefts. These two terms we'll discuss shortly and of course more extensive in a bit. Neurotransmitters are the structures, the molecules that are responsible for the either excitatory stimuli or inhibitory stimuli. And of course, the synaptic clefts is just the points where the neuron to neuron meet, or just any point of the ending of one neuron to another structure. It can be a cell, a neuron, nerve cell, or a muscle cell, or many other types of cells. We'll discuss them in just a bit. Before we do that, before we go on to this specific descriptions, let's first off see the neurons, and how they're actually structured. Well, first off, we distinguish the perikaryon, the cell body. Perikaryon actually means peri means surrounding, and karyon is the nucleus. So this is simply just the area, the main cell body, let's say, of the nerve system, and we can find within this the nucleus, the nucleolus, of course, inside the nucleus, along with all the other mitochondria and the nissl bodies. Now, what are the nissl bodies? Nissl bodies is just a special term used to describe the RER of the nerves, the rough endoplasmic reticulum, and this is because in the nerve system we find this unique staining method, this nissl staining method, and because of this, because we can actually see the granular appearance of the RER through this nissl staining, we also call the RER of the nerves also a nissl body. So this is the first and most important structure we can find, the perikaryon. Then we have the dendrites, these surrounding structures right here, these cellular processes, and they serve one function: to receive stimuli, to receive electrical signaling. So this is just the receiving structure. This is not a structure that propagates or generates or plays any role within the generation of an electric potential. This is just only to receive different stimuli from one nerve to the other, or one nerve to another structure. So this is again the dendrites. The term dendrite actually comes from the word tree, and this is because these dendrites look like tree-like processes, like branches and branching out of trees, and hence the name was very accurate. So after this we have the axon. Now the axon, this is the part of the nerve cell that that transmits also the electrical signals. Now the difference is, of course, just in the beginning, morphologically speaking, the axon is way thicker than the dendrites, and we have this axon halo, this special area that allows us to visually distinguish dendrites from axons, because the dendrites have directly very thin diameters, but the axon actually has a gradual shortening, a gradual narrowing of the this process, and allows us to more easily visualize which is which. So again, the function of the axon, of course, is to generate and most importantly to transmit the electrical signal, but also to transmit neurotransmitters, to propagate from one end of the cell body towards the very ending of the synapse this many unique molecules called neurotransmitters. We'll discuss in a bit what kind of substances can be considered neurotransmitters. So along this line, of course, along this axon procedure, this cytoplasmic procedure, we find many neurofibrils, which are again cytoskeletal parts, cytoskeletal components that allow this transmission of granules and other macromolecules and other molecules from the axon towards the synapse. Now the last point that we should see is the terminal synapses right here, the ending of the axon, which is again the ending of the where the electrical signal is going to be propagated from one cell to the other. So again, these synaptic clefts, we'll find out that they're also called boutons, also called buttons in the French, and the reason is this is because this configuration of the ending allows for a big surface area to be attached from the one cell to the other and allow a higher and faster propagation of the electrical signal. Of course, whenever we talk about the electrical signal or action potential in nerves, we simply mean this propagation of different electric voltage. There is the resting potential and the potential that is after the excitation, the action potential. So the resting potential is negative in the beginning, and whenever the nerve receives an input or receives the stimulus to excite itself or to actually produce and generate a potential, then we have an increased amount of income of sodium increasing from the negative voltage to the positive voltage. So this change from negative to positive, this is what actually means that the electric signal is present. This is the electrical signal when it comes to the nerve terms. This change from negative voltage to positive voltage. Of course, this has to be done. This is done through the balance of sodium and potassium. Typically speaking, we have the high income of sodium to excite the cell, and then we have the resting potential, which is the reverse, then restores the natural balance, and of course the balance to reach the resting potential yet again after the excitation signal is being produced. So these, of course, we can see also in the pictures, these axons, these nodes, we can see this myelin sheath. Now this is a unique feature in nerves. We'll discuss it in a bit, along with the cells that produce it. Just keep this in mind. This is called also the myelin sheath. So we discussed the perikaryon, the dendrite, and the axons. This is, of course, a microscopic picture to easily distinguish the nerve. And here we can see the dendrites, the narrow, of course, the directly narrow diameter of the dendrites, and we can see again the gradual narrowing of the axon, and this is also called, this area is called the axon hillock, of course. So let's focus a bit on the just the axons a bit more when it comes to the sheath. Now this myelin sheath, what is this? This is nothing more than just a cell product, again specifically from Schwann cells. Schwann cells produce in the PNS, and the oligodendrocytes produce the myelin sheath in the CNS. We'll discuss these cells in a bit. The point is that this myelin sheath is nothing more than aggregation of lipids, and the point and the purpose of this myelin sheath is to simply speed up and accelerate the nerve impulse, the nerve propagation from the axon towards the ending. And this happens because the nerve allows the signal to go in a halting mode, in a jumping mode from one node to the other. This neurofibril node is simply called the opening, the spaces between the different myelin sheaths surrounding the axon. So this allows the cell, of course, first to minimize the electrical losses, because of course, typically speaking, in the human body, we have the loss of the loss of electrical signals throughout the whole length of the axon, and of course, this is one of the reasons why the myelin sheath is present there, to minimize these losses, electrical losses, number one. And the second and most important is to accelerate the electrical signal, then the passing from the axon to the very ending of the synapse. So again, the electrical signal, the electrical propagation looks like a jumping motion, a haltic point of movement, and allows a jumping point from one node to the other, hence increasing the speed massively when compared to an unmyelinated axon and unmyelinated nerve. So again, in the end, we find the synapses, as I said before. We have this is the cleft, and the postsynaptic neuron, or this postsynaptic, let's say, cell, can be either a neuron or different other cells. For example, a muscle can be also one of the receiving end of the synapse. So let's talk a bit about the neurotransmitters. As I said before, the neurotransmitters are the molecules responsible for this excitation or the inhibition of this signal. Now, the neurotransmitters are a very wide range of molecules. They can actually be typically as simple as acetylcholine, which actually is used in the muscles, we saw before in the muscle video, and of course we have many other molecules. For example, the neuropeptides, or the amino acids, or the monoamines. Of course, this is a very wide range, and of course all of these molecules are produced in the cell body and propagated from the axon hillock towards the very end of the synapse, and then they actually are being exocytosed to induce this to either start or generate or inhibit or excite a general electrical signal. So let's talk a bit about the type of synapses. Of course, there's the axosomatic synapse, which is the axon, a nerve to a cell body, for example, let's say a muscle nerve. There's the axodendritic synapse, which is the propagation of the electrical signal from a nerve to another cell, to another nerve again, but only to receive. And this is simply just the receiving point of from another nerve, the dendrite, of course. And here we find a very unique structure called dendritic spine. This is a very important structure, and we'll discuss in a bit why the dendritic spines are very important. And lastly, we have the axoaxonic synapse. This is simply, again, axon to axon, and the point is, the point of this synapse is to moderate and to modify this this signal. For example, let's say that this axon propagates an excitatory signal, and this axon can actually modify through either also adding to this excitatory motion, again practically meaning providing an excitatory neurotransmitter, or an inhibitory transmitter to simply moderate or modulate the end result from the effector cell. So we will see different terms such as multipolar, bipolar, pseudounipolar, and unipolar neurons. Well, the term polar, of course, refers to how many of these processes go from how many of the cell processes exist in one cell. For example, the multipolar neuron receives many dendrites, has many cytoplasm that receive, of course, the stimuli, and the axon that propagates them. In this way, we have many polar ends and many polar ends receiving and one polar end giving, of course. The bipolar, of course, in this case, which simply means that there are just two endings, two different, let's say, sources of income. And in this case, we have the one source on the dendrites, and of course, the one common cytoplasmic process that is before this branching out of the dendrites. This is the one polar end. This is why we call it bipolar, one here and one here. And this is simply because the cell body, the perikaryon, receives only from two different points, nerve signalings and nerve formation. The pseudo-unipolar means that, of course, the pseudo always means false, always means fake, and in this case, because we can only see only one cytoplasm process, which again branches out, of course, to the terminals, to the dendrites, and again to the axon again. So this, even though it seems that it's only one cytoplasm process, again, this is a false image, because in between this area right here, the cytoplasm, there's the branching out in this manner. And last, we have the unipolar neuron, where only one cytoplasmic process leads to the perikaryon, and this serves both as a receiving end and as a giving as an end of provision. Now, this is the point why I wanted to discuss dendritic spines and why it's very important. Well, as I said before, the nerve has limited potential for regeneration. though our human body is designed in a way that it has to maintain vitality and function no matter what. So in case of an injury or some sort of necrosis of the cell, or any source of loss of neurons, our human body has to be able to somehow compensate for this loss. And as I said before, the death of a neuron is almost always permanent. There is no potential to regenerate or to repair this neuron whatsoever. So the point is that instead of actually producing new neurons, our human brain simply does different connections. We have, of course, the brain is filled, also the CNS is filled with interconnecting neurons continuously and continuously and all the time. So whenever there's a death of a group of the cells or area of these neurons, then the human brain tends to create again many synapses to simply compensate for this loss of the synapses. So the point is that the nerves, the alive and remaining nerves, tend to create more dendritic spines. Again, the dendritic spines are the endings, this embulgings in the dendritic areas. And this is, of course, all of these dendritic spines allow for other neurons to practically grab on. So the axon of many neurons can grab on back to this dendritic spine to create a point of connection. This whole procedure, this whole potential for re-establishing many points of connection and re-establishing the neural connectivity is called neural plasticity. And this is again the whole phenomenon and the whole ability of the human body to recreate many synapses just by using the old nerves and increasing the points of connection from one axon, from one nerve to the other nerve. And again, this is the compensatory mechanism after the loss of a neuron. So one last feature that we should discuss when it comes to neurons is again, the neurons are long-living cells. And of course, we already discussed in the heart, in the muscle video, that there is one common characteristic between the heart cells and the nerve cells. Of course, they're both long-living cells, and as a consequence, they typically, all long-living cells tend to contain tertiary lysosomes, or also called lipofuncin pigments. And this is the typical characteristic of the long-living cell. And this is this yellow-brown pigment. This is a lipofuncin pigment, and this is also typically found in long-living cells. Now let's start focusing about the neuroglia. What are all these supporting cells, how do they work, and what's their function specifically? So first up, we discussed about the myelin sheath. Again, the myelin sheath is just this covering filled with lipids in order to insulate mechanically, thermally, and electrically the axons. So in this case, the cells that produce the myelin sheath in the CNS are the oligodendrocytes. Now again, oligo means few, dendro means tree, and cytes is the cells. And in the PNS we have the Schwann cells. Let's see them, how they look like. Well, of course, this is the typical characteristic morphology of the oligodendrocyte. Again, this branching out procedure from many different axons, and again to produce this continuous coils of lipids around the axons in order to again increase the speed of nerve propagation, signal propagation, and of course to minimize electrical losses. This is a typical morphology of the oligodendrocyte. Now, in the Schwann cells are in the PNS, the peripheral nervous system. So whenever they are close to the axon, they typically tend to make this continuous coiling production of the myelin, and again, eventually in the end, they're going to have many layers surrounding the axon, and the nucleus is going to become punched out. Even though in the beginning it's again typically peripherally shaped, along with throughout the whole procedure and the production of many overlapping layers, this nucleus is being punched out and has this appearance of the, let's say, squamous-like appearance of the nucleus in the periphery of the Schwann cells, in the periphery of the axon. Now, let's meet one very important cell, the astrocyte. This is again a neuroglial cell that is the most numerous cell in the CNS, and actually they play a role in many functions which are very important. In fact, they are vital to the survival of the human brain. So first of all, they regulate the ion concentration. How do they do this? Well, this is because the whole rationale is the location and the strategic placement of the astrocyte. Because the astrocyte is the connecting cell between the vascular supply, the capillaries, and the nerve system, this places them in a position where they can actually regulate everything that goes from the blood to the interstitial tissue and the neurons themselves. So in this case, we can actually see that because they cover up the capillary of the nerve in the nervous system, they actually selectively allow ions to go from and to the interstitial tissue of the CNS. So this is the first very important function of these cells, to regulate ion concentration. And this has as a consequence also responsible for the electric potential, because nothing more than as we said before, the electric signals is nothing more than just an ion exchange from sodium to potassium and so on and so on. So this is a majorly important role when it comes to the ion concentration of the interstitial tissue of the CNS. Second function is that they guide and support the neurons. This is again a supportive role as we discussed before. And again, here we meet the first very apt example of the blood-brain barrier, of the blood barriers between the one specific vital structure and the vascular supply. So we'll discuss in just a bit what is this blood-brain barrier and how does it work. So far, just have an idea that this is the connection between the blood-brain barrier is a selective filtering structure that allows only specific molecules to go in and out of the blood towards the interstitial fluid and the interstitial area of the brain. And lastly, we have the function which is to regulate vasodilation. This is important. Why? Because vasodilation, vasoconstrictions is the one way that the cell can control the amount of nutrients, the amount of oxygen coming from and to the structure, to the interstitial spaces, and of course in the cells. So this is very important when it comes to the functionality and the vitality of the tissues. Now, another supporting cell is the satellite cells of the ganglion. Now, the ganglia, as again said before, this is again the aggregation of many nerve cell bodies or many nerve cell procedures, cytoplasmic processes. And of course, in this case, the satellite cells are supporting cells, trophic. Trophic means that they actually provide nutrients to the cell. And of course, the satellite cells are the cells that surround the ganglia after the spinal cords, the nerve and the spinal nerves. So again, this is how they look like, and we're gonna see them in the microscope in the nerve system in another video. And at this point, just to understand, this is a ganglion again cell, a neuroglial cell, and the point is to support and provide nutrients to the cell body of the sensory neuron. One of the last point cells is that we should discuss is the microglia. Now, the microglia again belong to the monocyte phagocyte, so it's mononuclear phagocyte system. Again, this is nothing more than just the macrophage of the CNS and the PNS. Here we find that all of the immune reactions when it comes to the infections or any sort of pathogen, any sort of foreign body, the neuroglial cells will perform the same function of the macrophages, which again are phagocytosis, inflammatory markings, pro-inflammatory markings, or anti-inflammatory markings depending on the phase and depending on the stage. And of course, this is nothing more than just the specialized macrophages and monocytes within the CNS and the PNS. Now, this ependymal cells is going to be the first step to a very big discussion when it comes to the CSF, the cerebrospinal fluid. So the ependymal cells are cells that are typically found in the choroid plexus. We'll discuss where the structure is in just a bit in the picture. And the function of these cells is to produce and move cerebrospinal fluid. So, the ependymal cells are typically cuboidal cells with centrally placed nuclei, and on the top they have cilia, specifically kinocilia. Now, these cells, of course, are responsible for the production of CSF. What is CSF? What is the point of the CSF? Well, this is just a microscopic picture of how these cells look, the ependymal cells look like. And this is, of course, our first step to a discussion of what is the CSF. So the CSF is nothing more than just a liquid that has the qualitative, the elements that are contained inside, the ions, the nutrients, and so on, are similar to the blood plasma in quality, not quantity. The blood plasma and the CSF have, of course, differences when it comes to concentration and when it comes to some components, but the specific molecules that exist within the CSF and the blood plasma are exactly the same. Different quantities, but actually the same molecules exist. So this is the choroid plexus right here, and this is the location where we have the ependymal cells, and again these cells produce CSF and they move it towards inside the whole system of the ventricular system. We have the lateral ventricles, third ventricles, fourth ventricles, subarachnoid displays, and arachnoid granulations. This is the whole procedure of how we produce CSF and how we also drain it. So there's a whole system in the human brain that allows the CSF to be present. We have specifically approximately 130 milliliters every time during the day of the CSF in the human skull, in the human brain, surrounding the human brain, and the function is we'll be discussing in just a bit. So there's a whole procedure of continuously producing CSF and continuously draining CSF. The production is primarily done through the vascular system and secondarily by the ependymal cells, and the absorbing and the resorption of the CSF is actually done through the venule system. Of course, we have this arachnoid granulations which go from the brain, from the meninxes of the human brain towards the vein system, and again they drain and they filter, of course, the structure, this CSF fluid. So let's discuss what is the function of the CSF. Why would we have a liquid throughout the whole day, throughout the whole our whole lives surrounding the brain? Well, this is because the brain actually is a very, very heavy structure. In fact, it's approximately 1.5 kilos fat. This is a quite big structure. This is a quite heavy structure, and because of the vitality and the necessity of the brain, of course, this organ has to be protected. So we have a problem because the skull is covered through is a very hard area, it's a very structured, very, very dense area, and whenever you have a 1.5 kilo of mass surrounding by hard surrounding, this is very, very easy to mechanically irritate and mechanically destroy the structure. So how would you protect a big organ of 1.5 kilo from hitting its own cell through it on the walls of the skull? Well, this is how the human body solves this problem. The CSF is again the liquid that surrounds the whole brain, and in this way we achieve buoyancy. This is again the effect that we see typically, that the mass, for example, our mass is exactly the same in the sea and outside the sea, but our weight is different. Our weight is different inside the sea and of course and outside of the sea. And exactly the same logic is applied in the CSF. Because the mass cannot be changed, the mass is mass and it's going to stay the same, of course we're going to encase and uncover the whole brain in this liquid that is going to change its weight to 25 to 50 grams. This is a major weight reduction that protects the brain from mechanical injury and actually safeguards it from mechanical injury throughout the walls of the skull. So this is the first and most important structure, this function of the CSF. Of course, also it actually also works as a shock absorber because it protects the brain from sudden pressure or temperature changes. Also, we find components of the immune system inside, and this is important. Why? Because in order to distinguish and to be able to take a sample from the brain, of course, to discuss if it's an infection, a meningitis, or any infection in the CNS, one very good way to take a sample from this, to visualize or to confirm or reject a possible meningitis, a possible infection in the CNS, we take a small sample of the CSF. This is the medical procedure that would simply just aspirate CSF liquid from the spinal cord, and we actually can analyze the components, and depending on the results, we can either confirm or deny or reject a possible infection. So this is also a very, very important thing to mention, absolutely. Of course, we also have the metabolic in the way that the CSF is one of the major ways of production of CSF is selective filtering of blood plasma. So I said before in the beginning, there are two ways. One is the ependymal cells that produce and move the CSF, but the major source is just a very, very selective filtering from the blood, from the capillaries, from the vessels of the vascular system towards the interstitial fluid of the brain. There is a very, very selective filtering. So in these cases, the endothelial cells and along with the blood-brain barriers and the blood-CSF barriers, they specifically filter and the blood plasma, and the blood plasma that goes from the vascular system to the space where the CSF is located is the actual product of the CSF. And lastly, there's an indirect function of the blood flow, in which case is this: because of the CSF, of course it's a liquid, of course, and all liquids and all structures exert a pressure. And specifically, this is going to be called the intracranial pressure. Cranium is the skull, intra inside the skull, the intracranial pressure. So the amount of CSF is somehow, of course, it's actually correlated to the intracranial pressure. So there is because of the amount of the CSF being present, this is an indirect, let's say, regulatory point of the intracranial pressure. So whenever we have high intracranial pressure, the CSF tends to be lower. This is just a simple sort of a compensatory mechanism from the CSF and the intracranial pressure. So let's discuss about these two structures I just mentioned, the blood-brain barrier and the blood-CSF barrier. Generally speaking, our human body has many barriers. You'll hear the term blood something barrier many times. For example, blood-air barrier in the lungs, or blood-brain barrier in the brain, or blood-CSF barrier between the blood and the CSF, the cavities of the CSF, the ventricular areas. So in all of these cases, either the blood-air or the blood-brain or the blood-CSF, we're going to find always the endothelial cell, which is again the lining, the cell that covers the innermost part of the capillary, of course. So it's going to be always the endothelium, the basal membrane, which is going to be common. The basal membrane is going to be again produced. There are two different layers of the basal membrane: the basal lamina and the reticular lamina. The basal lamina is always produced by the endothelium, and the reticular lamina is always produced by the cell that is beneath it. So, for example, in the blood-brain barrier, we have again the endothelium with the tight junctions in between them. This is again our filtering points, of course. All of this area, all of this barrier is a filtering point. So this is the first boundary, the first limit of on the molecular basis of the barrier, and this is the tight junction between the endothelium. Exactly beneath it we have the basal membrane, which is the fused basal membrane from the endothelium and the astrocyte. And again, the basal membrane is nothing more than, it's again the typical structure we discussed, and because of its ionic charge and because of its conformational characteristics, this also functions as a barrier for the blood-brain barrier. And lastly, we have the astrocyte. We discussed about the astrocyte. This is again the cell that has attached, and these processes of these cells are attached throughout the whole basal membrane, and again selectively filter molecules coming from the blood to the brain. This is the first example, and of the blood barriers, generally speaking. And the second we're going to see right now, the blood-CSF barrier. Again, this is similar, but there are tiny differences. The endothelium, again, in the blood-CSF barrier doesn't contain tight junctions in the same way that has also the blood barrier contains. Here we have the choroid cells. Again, these choroid cells are again the ependymal cells. Now, the choroid cells are more responsible for the specific filtering of this case through the tight junctions, and of course through the active movement of ions from the blood towards the cerebrospinal fluid. These ions, of course, include sodium, potassium, calcium, and so on, and many others. Now, let's just discuss the correlation between the gray matter and the white matter, and how does this correlate to the histological points. Well, this is because in this case we can see that the gray matter is nothing more than, it's called gray matter because there is no fat, there is no myelin. And because of the white typical feature and the whitish yellowish hue of the fat, this is why we call it gray matter. And of course, inversely we can find that the white matter is nothing more than just many areas, many nerve fibers that are myelinated. And of course, this whitish yellowish color of this hue of the fat is the one that gives it this appearance of the white structure. So let's see an example of the neurons. For example, we can find that in the cerebellar cortex. Now, in this case, the cerebellar cortex, just shortly, is made out of two different layers: the cortex and the medulla. Generally speaking, always the cortex is going to be the area that is on the outer part, and the medulla is going to be the inner, more central part. And of course, we can see the medulla actually extends in the same way that the cerebellum extends. So the cortex is subdivided into two layers, the molecular and the granular layer. At this point, this is just a distinguishing, I'm just mentioning the structures just for you to understand where we can find these Purkinje cells. Careful, do not confuse Purkinje cells with Purkinje fibers. Purkinje fibers are specialized cells in the heart, and the Purkinje cells are specialized cells in the nerve system, completely different cells. So, let's zoom in actually and see them. In between the molecular layer and the granular layer, exactly on the top of the granular layer, we're going to find these very, very typical pear-shaped cells. Again, this is a very characteristic appearance of the Purkinje cells. Let's visualize them also in histology guide. So again, this section has the brain, and this is the cerebrum, and exactly on the right part, this is the cerebellum. So if we zoom in exactly and focus on the in between the layers of the molecular and the granular layer, we should be able to visualize and find the Purkinje fibers, Purkinje cells, sorry. So there we go. We can actually easily identify the very, very large cytoplasm of the Purkinje cell, and specifically there's another one, and another one is right here. And of course, all of the other cells are typically either other neurons, or typically, statistically speaking, they should be the neuroglial cells. Also important to mention is that the population of between neurons and glial cells is again higher. The neuroglial cells are way higher in amount than the neurons themselves. So this is again a typical characteristic and typical location and presentation of the Purkinje fibers, Purkinje cells, sorry. So just to want to point something out, that the processes, the cell processes of the Purkinje cells are actually extending from the granular layer to the very, very end of the cortex. Their cellular processes are very, very long and extend again from the very top of the molecular layer to the very, very top of the granular layer. Thank you very much.